I'm going to end my model. So this is class 6 and this is class, my model will get over. So for my model, I'm going to have a stage in the next week during the tutorial time. So couple of you still have not got registered for the thing or the tutorial because the registrations are coming so late, and at a times I don't get the updated information from the ASC. So by the time of the next week, I think hopefully all those issues will get resolved. Still, some of you are contacting me that you are no, you have not got the access for the Zoom and all other things. So if at all, if you have your friend facing some kind of problem, then I would say just share the email with that person so that the person can get registered. But during registration only IIT Bombay email address will be allowed. Okay, so coming to today's lecture. So today's learning objectives are first basically about sales cycle. Kabir, I have told many times whatever the questions are there, I will answer those questions at the end of the lecture. I cannot have this interruption at this moment. So just hold on until end of the lecture, and then at that time you can ask me. So about the cell cycle, particularly what we'll learn is mitosis, meiosis, and the checkpoints. How exactly cell will, there are some signals, and those signals have to be followed, and then there are checks and balances to have the cell cycle. And then we'll talk about structure of DNA and few preliminary experiments which were done in 1960s and 70s in order to understand the DNA structure and the replication pattern, in order to understand how genetic information is passed from one generation to generation. So when you think about cell cycle, again, intuitively what you think that cell will grow and there will be some kind of growth, and those growth are related to something. And in this context, in cell cycle context, mainly we are going to talk about the nucleus, or the instead of nucleus, I would say we are going to talk about the DNA or chromosome. And the chromosome basically during its interface. So when you think about the cell division and its role, so the cell division will distribute the DNA of mother cell equally between two daughter cells, thereby allowing to have cell to proliferate. And this basically cell proliferation is very essential in terms of its growth, repair, and reproduction. So when you think about cellular organization, and when you think about the information, how information is passed from generation to generation, the most important molecule in that context is DNA, or one can say genetic material. And when it comes to the genome, for the eukaryotic cell, you might be able to see that there are number of DNA molecules, while in case of prokaryotic cell, there would be only single DNA molecule. And then in that context, in cell division context, this DNA molecule is called as a chromosome, and we will talk about that chromosome in subsequent slide. So in terms of cell division, we have to understand two particular terminology. First is somatic cell, other is gamete. Now, when you think about somatic cell, the somatic cells are non-reproductive cells, and it has two sets of chromosomes. However, when we think about gametes are reproductive cells, and if you think about reproductive cells, then you can think as a sperm, and egg, ovum, and these chromosomes has the gametes has only half of the chromosome as somatic cells are there. So somatic cells in that way, let's say you have two N chromosomes, then the gamete cell will have only N chromosomes. And coming to the cell division, when you think about cell division, there are different stages. And the most important or the longest phase you can see is the interphase. And during the interphase, the cell will have the growth and it will have lot of metabolic activities. But then, since the growth is there, even the growth is, there is a different time, there will be different growth. So, according to that, the interphase of the cell cycle is divided mainly into three parts or three divisions. G1, then basically growth related cells, or can say first a gap or G1 phase. And the second is basically synthesis phase, and the third is G2 gap or second gap. So the synthesis phase mainly important because over here, not that even G1 and G2 phases are important, but in this cell cycle context, the S that stands for synthesis, and this synthesis is mainly towards the DNA, and that is why the S phase or H phase is a little bit important over here. And then during the S phase, there would be a lot of duplication of the chromosomes will get duplicated, and in the G2 it would be the last phase of the interphase, last part of the interphase or last phase of the interphase. And after this G2 phase, the cell will get into division. And that division, depending upon its somatic or its reproductive cell, that would basically it's either mitotic or mixed. But in this case, it is a mitosis. We are talking about at this moment somatic cell. In mitosis, basically there is a cell division. The chromosome will get distributed to the daughter nuclei or daughter cells, and that is called as end cell. Mitosis, that is end cell. And after mitosis, it is basically cytokinesis. Cytokinesis is basically dividing cytoplasm and producing two daughter cells. So in cytokinesis, as you know, you have basically if you have somatic cells during cytokinesis, you will have two daughter cells. Now, when you think about, let's say, you are thinking about the human cell, and let's think that the cell cycle or one division of the cell cycle that happens for 24 hours. And if you consider this 24 hours, then among this 24 hours, approximately 5 to 6 hours are being spent in G1. Approximately 10 to 12 hours are being spent in the DNA synthesis or the S phase, and again 5 to 6 hours are spent in G2. So, and then when you think about mitosis, the mitosis is really relatively, fast, and this mitosis happens on the scale of 1 hour also. So these are approximate time scale, and depending upon organism, the cell cycle, the duration will vary. But for the typical human cell, is basically, you can say, or you can estimate that this particular one division happens within, let's say, 24 hours or so. Now, there are two types of cell cycle. One can say mitosis, other is meiosis. And when you think about these two, there is a peculiar differences over here. In mitosis, again the division is for only somatic cells, and it will have two daughter cells which will have the same amount of DNA as mother cells, or so basically same amount of chromosomes. However, in case of meiosis, the division is for the gamete cell, or you can say it's a sperm or ovum cell. And in case of meiosis, there would be 4 daughter cells. However, in this meiosis case, each daughter cell will have only half of the amount of DNA as mother cell. And in detail, we will talk about mitosis and meiosis, how this division happens, and what is basically in that order. What do you mean by same amount of DNA as mother cell? How exactly that happens? So when we think about the mitosis basically produces new cell, and it has a capability to replace the old cell, or it can basically replace the damaged or lost cell. And most importantly, the mitosis will have a division of 2 identical daughter cells. This is the typical mitosis picture of cell fluorescence microscopy. What you see, these are the microtubules, and what you see over here, these are the 3 chromosomes or chromatids. As I know, sister chromatids, and these are 2 poles which are held by centrosomes. And it's a very beautiful picture. Now, before getting into the basis of the mitosis and meiosis cell division, we will understand few terminology. Okay. So, it's a basic. So, when you think about the chromosome will have a pair of sister chromatids, and each chromatid will look like this. And chromosome is basically something called as centromere at the center of it. And with that centromere, both chromatids are held together. So basically, each chromatid, when you think about the difference between chromatid and chromosome, as such there is no big difference. But chromatid, what you can think as, it's a loose form of DNA. And basically, the DNA is in the chromatin format, chromatin form. The DNA will be loosely, wrapped with loosely, attached protein, you can say. And when you think about chromosome is basically very compact form. So the chromatid will further get into assembly, and then it will have compact form. There will be more proteins coming there, and because of that, it will achieve the compact form. And when you think about chromosomes will appear only the time of the cell division, as a meiosis or mitosis, and the other time in the interphase, basically most of the time interphase, DNA will be in the chromatid form, because there are a couple of factors involved over there. The DNA, what you think is, it is a very long, you can say, polymer, and on the DNA you have a lot of genes. So in order to have a particular metabolic activity, that gene has to be located, or that gene has to be accessible. And in order to have that accessibility, couple of sites on the DNA will be open, always open. And that's basically very well, and that is the significance of these 2 forms of the DNA. Again, the compact form, you can say that is a chromosome. The DNA's compact form, that would be a chromosome, and the loose form, that would be a chromatin. And within a chromosome, you have the sister of chromatids, a pair of sister of chromatids. Now, when we think about the haploid versus diploid cell, or when you think about the DNA material of the parent cell, in chromatic cells also, what you always find in chromatic cells, that it's a diploid. What does that mean? The diploid means it has a set of chromosomes from the maternal as well as same set of chromosomes will be from the paternal side. And that is why the case is basically over here with 2n. So the one n is coming from the maternal side and the other n is coming from the paternal side. Now, depending upon the organism, number of chromosomes will be different. In this particular diagram over here, what you see is 3 chromosomes are from the maternal side and 3 chromosomes are from the paternal side. And that is why there is a duplication, and what we call this the same chromosome. So but then the same chromosome is basically one chromosome is basically from maternal side and other is from paternal side. So this is called as pair of homologous chromosomes, which is basically one from each set of maternal side and set of paternal side. So and then there are total 3 chromosomes. You can say pair number 1, this one, pair number 2, and pair number 3. And again, here the basic structure is like this. You have pair of systematics and all those. Now, in case of human chromosome, these are you can see these are the human chromosomes, and each human chromosome, then you have duplication, or you can say it's homologous duplicated chromosomes over here. In this each picture 1, 2, 3, 4, 5 and so on until 22 also you have set of homologous chromosomes, and each chromosome you can see there is a centromere and then there is a pair of chromatids or piece of chromatids. Now when it considered for the male and the 23rd chromosome, that is different in case of male and female. In case of female it is XX and in case of male it is XY. And how exactly that this gene is passed from the parent to the fertilizer, that would determine the sex of the offspring. So in this case particular, what we can say, there are 46 human chromosomes, but these 46 human chromosomes has 23 types, and each type, when you think about each type, it is nothing but the homologous duplicated chromosome. In case of diploid form of the cell, diploid chromosome, you will find a homologous duplicated chromosome, and in case of haploid form, it will be only one chromosome. Again, I am repeating this. In diploid format of the cell, you will have a pair of homologous duplicated chromosomes. So, and in the haploid, there will be only one chromosome, and that is the main difference between haploid and diploid cell. Now, when we think about the cell division, particularly about the mitosis, it will start from the G2 interface. And in the G2, what would happen is the cell is, you can say it's matured. There is a centrosome over here, and the nucleus. Inside the nucleus, you can see a lot of chromatids, and it's it's in chromatid form. The DNA is not compact form, and it will only achieve the compact form in the during the cell division. So the first phase of the cell division, or first phase of the mitosis, is the prophase. And in the prophase, what you see is the centromere, it starts, having, getting into some distance, and we will see there is a development of this mitotic spindle, and this spindle is nothing but microtubule structure. And then at the same time, what we see, the chromatids are being, the chromosome is being appeared, and the DNA is getting into the compact form, which is nothing but chromosome. And now from this point to this point, interface to the prophase, the clear difference is there is a appearance of the chromosome. And in next state, that is pro-metaphase also, over there what would happen, the nuclear membrane over here, this nuclear membrane of this thing, that would disintegrate. And when that disintegrates, then the next stage will achieve, and then in the pro-metaphase to the metaphase will have start. And in the metaphase, in the metaphase, what happens is this centromere, you can see they are at different poles, and what happens is there is some kind of plane over here, and this plane can be called as metaphase plate. And what you see in this metaphase plate, all these chromosomes will get kind of arranged in that particular plane, and each chromosome is basically attached with the spindle fiber. In the next phase, anaphase, what would happen is, this is, remember, this is a single chromosome, and this single chromosome has a pair of chromatids, and that pair will get split, and now there will be a chromosome with only single chromatid, and that basically in anaphase, what happens is this chromosome starts getting pulled to respective centromere, and what would happen is eventually the spindle fiber will get dissolved or disappear. It will get depolymerized in the telophase, and in the telophase time, what you see here is the nuclear formation starts. And then at the same time in the cytokinesis, what would happen is there should be some kind of cleavage through that would indicate the division of the cell. Now, in the cytokinesis particularly, as I said, there is a cleavage through, and with that it would start the basically division process, and eventually this cell will get divided into two daughter cells. So in case of mitosis, what we see is over here the number of the cell, each cell is a diploid cell, and then you have the daughter cells. Basically, two daughter cells will be produced. And this is a movie where we can see the mitosis happening. At this moment over here, it's the prophase, or just after the interphase also, and the chromatid is basically getting aligned. The cell division, basically you might be able to see some kind of formation, and then after some time it's a metaphase. In particular metaphase, what you see is the chromosome starts get aligned and become plane or metal, and then the centrosome starts appearing, and then basically it is basically there is some kind of attachment over here for each chromosome, spindle fibers through spindle fibers, and this is somewhere over here. About this time you can see some kind of spindle formation, and it is starting to get into some kind of phase, and now the chromosomes all of a sudden you will see it's getting pulled up. At this point, this is the onset of anaphase, you can say. And from this point, you see the chromosomes are getting polarized, or it is basically moving to each pole, And then you will see there is a cleavage formation over here. This black line, that is a cleavage formation. And this is the fluorescence movie in which again you can see the cell division or the nucleus. You can see these cells over here, anaphase. This is anaphase. And after that, once the chromosome starts pulling at respective pole, the spindle will get depolymerized. I'm going to play this movie again so that you can watch it. And below is the link. So until this point, it will be prophase, then starting of the metaphase, and now it will be I just want anaphase, and then when you can see this line coming in between, the black line, black dot, yes, this one is the diffuse telophase. And telophase, after that, you have the splitting of this cell. So now we will talk about this was about the mitosis. And again, remember in mitosis, we have two daughter cells, and each daughter cell will be diploid. Now we will talk about meiosis, and first we will understand what is the importance of the meiosis. So basically, meiosis is very important in terms of generating the genetic variation in the offspring. And particularly in the meiosis process, there is a term called shuffling of genes, and that shuffling happens across the homologous chromosome. And due to that shuffling, what we will see that particular characteristics of the the kid might have particular characteristics of either mother or father, and that is because of this shuffling. And when we think about the meiosis, it's very, you can say very important in terms of the reproduction. And when you look into this diagram, and when sperm and egg basically gets into fertilization, then it would be having a diploid cell. Otherwise, sperm and egg will have only single chromosome. There is no pair. And when it undergoes into the fertilization, then it would be a zygote. So before that, each haploid gamete or the gamete has only 23 chromosomes. And in the diploid form, it would be 46 chromosomes. And then basically there is a meiosis and all those things happening, and with that we will get into the further cells. So diploid cells will have two haploid cells, and the chromosome threads in the zygote, both chromosomes will be in the zygote, and all these genes they will carry to the very precision, to the somatic cell. But the most important point over here, you want to understand that in the haploid form you have 23, and in diploid form you have 46 chromosomes in case of human. Now this is how the cell division happens in the meiosis process. Okay. So in the meiosis, there are again two stages or two steps, or you can say two divisions: meiosis 1 division and meiosis 2 division. In the meiosis 1 division, basically what happens is haploid cells with duplicated chromosomes are there. Otherwise, what you can say about meiosis is that it will have homologous chromosome separation. So it will have the haploid cells with duplicated chromosomes. And in meiosis 2, particularly this is basically you have at the outcome at the end you have haploid cells with unduplicated chromosomes. Now let's try to understand it. So in the meiosis, as I said, there is a particular term which is called as crossover, and that crossover happens in the homologous chromosome. So in particular over here, these are some points and that is called as chiasmata, and with that those particular points, what happens is the gene from this side, the red, that would get into the blue gene and vice versa. So that is called as a shuffling or crossing over. And with that variation, each offspring will have a different characteristic little bit. The overall, the appearance will be same, but then the appearance, even though their appearance, there would be peculiar difference. And so basically, this is the prophase one. Again, in the prophase one, what you see, little bit spindle formation, the centrosomes are basically getting apart. And in the second stage, that is metaphase, what you see is the homologous set of chromosomes that is basically attached to the spindle fiber, and they are together. And then the centrosome, they are basically aparted, and they are at different poles or exactly opposite poles. And then what would happen is that is the metaphase, and in the next phase is the anaphase, and at the anaphase the main difference over here is from the metaphase is this chromosomes, they are basically being pulled away. But now you look into this chromosome. So this chromosome is basically, you can say, it's a pair of chromosomes over here. And then there is actually in the next phase you have the telophase 1 and cytokinesis. Now in the telophase, again you will see this cleavage happening between the two cells, and there will be some kind of a nuclear structure over here. The spindle fiber that will get disintegrated or that will get depolymerized, and then there will be two daughter cells at the end of meiosis 1. But these two daughter cells are in the haploid form. What it has, chromosomes. Each chromosome consists of two sister chromatins. So basically, what we are getting here is two haploid cells, and each chromosome consists two sister chromatins. Now, these two individual cells will get into the second stage of meiosis, that is called as meiosis 2. And in meiosis 2, again you have prophase, metaphase, anaphase, and telophase. Now over here, again in the prophase, what you see, the centromere, basically centrosome will getting aparted, spindle formation will be there, and the metaphase, again this chromosomes will get arranged, this metaphase plate, and then in the anaphase the chromosomes are getting aparted. Now over here in this over here, you can see the sister chromatids are getting separated, which is similar like anaphase of the mitosis. And then the next phase, basically you have the telophase and cytokinesis. And in telophase, again what you can see is the spindle is getting disintegrated or depolymerized, and the centrosomes are basically each cell will have one centrosome, and so. And then each cell will be divided into two. So at the end of meiosis, you will have 4 daughter cells, and most importantly, each daughter cell will be in the haploid form. Now, when we, here I'm going to show you one movie about meiosis. I'm just going to fast forward. Okay. So this is the event where, first event over here, you can see this is the, okay, this is the anaphase also of meiosis 1. And then there is a division. You can see there are, two cells, kind of, cleavage forming happening over here. And you have, so now the two cells are here. And now these two cells will further get into splitting. Now over here, this particular point here, this is the anaphase, and from this point, you can see there is a separation of the chromatid, and now it is getting into 4, and then there will be 4 daughter cells. I'm going to play this movie again so you can see it clearly. Okay, so when we think about the mitosis versus meiosis, there are, sometimes it's very different things. So in particular, there are, I can say, 3 distinct, points. The first point is the crossing over. Over here, during the prophase itself, prophase 1 of meiosis, the crossing over happens, and in that crossing over, what happens is in the homologous state over here, you can see there is an exchange of gene in between the homologous chromosomes. So that phenomenon is not there in the mitosis. Okay, and first of all, you have to understand that there is a homologous state over here. One state is coming from the, each state is coming from one parent. So that is, very peculiar relationship over here. And then when you think about the homologous pair at the metaphase plate, in the metaphase 1 of meiosis, chromosomes are positioned at metaphase plate as pairs of homologs. Okay, so basically this is pair of homologs in the metaphase. And in this mitosis case over here, this is individual chromosome, but here it's a pair of homologous chromosomes. So that's the difference. And this metaphase over here, that is very much similar into the second phase of the meiosis. And then, so that is, one of the distinct points. And then the third point over here is the separation of homologs. So in the anaphase 1 of the anaphase 1 of the meiosis, the duplicated chromosomes of each homolog pair will move towards the opposite pole. And the sister chromatids of each duplicated chromosome remain attached. However, in case of mitosis, the sister in mitosis, you can see that the sister chromatid gets separated. So that's one of the differences you can say during the mitosis and meiosis. Now, turning to few similarities versus differences in mitosis and meiosis. When we think about DNA replication, in both cases that occurs during the interphase, which is before the cell division, either through the mitosis or meiosis. And in case of mitosis, there is only one time cell division, and with one time cell division, you can have 2 daughter cells. In case of meiosis, there is 2 times cell division, and with that, you will have 4 daughter cells. When you think about the crossover or synapsis of homologous chromosome, it does not happen in the mitosis. and in contrast to that, in meiosis, it occurs along the crossing over between non-sister chromatids in prophase 1. So, and again, when you consider the how much genetic information or how much DNA is basically passed from the parent cell to the daughter cell. So, in mitosis, you have two diploid cells that are basically genetically identical to the parent cell. However, in case of meiosis, you have four haploid cells and each containing half of the chromosome as parent cell. And each daughter cell is basically little bit different from the parent cell. And also because of that particular crossover or the synapsis phenomenon, synapsis of homologous chromosomes, because of that, each daughter cell will have little different. But this could be little different in case of identical twins, where but then we are not going to talk about it. But even in twins, it could be identical or non-identical. In non-identical, it is basically a 2-egg formation, and in identical, it's 1 egg which is getting divided into 2. So it carries same genetical identical twins. It will have same genetic material. And when you think about the role of this mitosis versus meiosis in the organism, so in case of mitosis, it will produce the cell for the growth and repair, and in case of meiosis, that is basically mainly towards producing the gametes. The gametes would be, you can say, sperms and ovum, and it assures genetic diversity in sexual reproduction. Now we'll talk about the checkpoints during the cell division and cell cycle. So when you think about the cell cycle, before entering into the prophase or before entering into the phase, there is a, you can say, G0, which actually G0 is non-dividing cell, right? So there are particular proteins inside the cell. They appear, they appear throughout the cell cycle, but then their concentration varies during different cells. And in particular, entering into the G1 cell, before entering to this G1 cell, these cyclic proteins, so because it appears cycle-wise, and that's why it is called as cyclic protein. So this cyclic protein will cross-check whether the cell wants to get into the cell division, and then it would get into G1 state or G1 section. So basically this is the first checkpoint, and this checkpoint is called as G1 checkpoint. And if at all cell receives the go-ahead signal, then it will get into the it will continue into the cell cycle. And before also, before getting into the mitosis, there is a particular state, pro-metaphase also. In that particular state, what will happen is cell will check whether each chromosome is attached to the spindle fiber or not. If at all it is not attached to the spindle fiber, the chromosomes are not attached to the spindle fiber, then it will not get into the mitosis, or it will not get into the cell division cycle. And that is also one other checkpoint, and that checkpoint is called as end checkpoint. And once it assures that basically each chromosome is attached to spindle fiber, then the metaphase will happen, and then it will get into the subsequent steps of the cell division or mitosis also. So these checkpoints are very much necessary in order to control the cell cycle. Otherwise, there would be cell division which would happen, and then there won't be as such control over genetic information. And in order to have that particular control, you need to have these checkpoints. Um, when you think about the cell cycle, it's just not basically these checkpoints which are being regulated. The cell cycle is only just it does not get regulated only by this checkpoint, but also it gets regulated by some other external signals such as nutrients, growth factors, and then the density dependent inhibition and substratum for anchoring. These are also some points or some external signals which required before the cell division has to happen or cell cycle has to go on or continue. So when we think about the cancer cells, so there are basically sometimes in cancer cells there are no checkpoints and they do not exhibit density dependent inhibition. So which is basically multiple layers of the cell. And they do not require the anchorage with the substratum. So over here and at the right side, what you see is the normal mammalian cell, and on the left you can see this is cancer cell. The structure is little different. Over here you can say the mammalian cells, they are having the growth by layer by layer. And in cancer cells, the growth is not layer by layer. You can see there is some kind of some cells are getting out of plane, and there is kind of obstruction. When you think about this cell cycle or cell growth over here, the normal cell growth will have this kind of structure. You can say it will happen plane by plane. And in case of cancer cells, some cells will protrude out of this control cell gate, control or regulated cell growth. And this is out of control, and that's why you can say it's a cancerous cell also. But again, the cancer cell has many reasons to be. It just not one mechanism. There could be many other mechanisms why the cancer cell growth happens. Now I'm going to discuss about the next meaning objective, that is about the structure of DNA. And over there, it's a very interesting history, and that's all history. I mean, the experiments have been done in 1960s also, 1960s, 1960s. So just before you start with this discussion, then I want you to remind how this DNA structure looks like. At this point of juncture, we know the structure of DNA, and that's why we can discuss it. But then, when the scientists started, they did not have any idea. But coming to this structure, you remember that there are nitrogenous bases. These nitrogenous bases are pyrimidine and purine. And in case of DNA, it is thymine, adenine, cytosine, and guanine, or ATGC, can say. The one letter code for adenine is A, for thymine is T, for cytosine is T, guanine is G. These are basically one letter code. And mostly in the DNA, whenever you you look into the DNA sequence, you will see that sequence is written in the single letter code. Now, this basically the nitrogen bases is attached to the sugar base, and in this case for the DNA it's a deoxyribose, because at this point this 2' position over here there is absence of this oxygen OH. This is not hydroxyl group, but just hydrogen attached over here, and because of that it is called as deoxyribose. And at the 5' end you can see there is a phosphate. So the phosphate for this structure, the DNA, it can attach to 3' end and 5' end. And generally you will see, so this is the 5' end and this is the 3' end of this DNA chain. So there is a directionality for the DNA, and they basically the nitrogen is there along the deoxyriboes and then along with the phosphate, that is whole as called as nucleotide. Now, coming to the initial experiments when the scientists started to understand what is this DNA, how it is made of, and what kind of composition it is. So one of the earliest experiments was done by Erwin Sergeant, and what he observed, so what he basically did is he took DNA from the different organisms and he tried to quantify the amount of this ATGC or adenine, guanine, cytosine, and thymine. And on that basis, he made some observations. So over here, what you see, adenine and thymine, they have a little bit, very, you can say, same composition. So this is 32.8% and thymine is 32. In E. coli, it's 24.7, it's 23.6 thymine. In human, it's adenine is 30.4 and in thymine it's basically 30.1. So they are approximately, you can say, same content. And same thing goes for guanine versus cytosine. 70.7, 70.3 for the fuel rich, and for the wheat 21.8 and 22.7. So these are very close numbers. So on that basis, what he concluded that DNA is a polymer consisting of these four nucleotides A, C, G, T, and in the first observation what he made is the base composition of one organism could be little different than the other organism. So, for example, when you think about human DNA has adenine as a 30%, while the E. coli, that has 24% of 24.7% of adenine. The second observation made by him is very interesting. So, what he found as number of adenine and number of thymine, they are same, approximately same, and same thing, number of guanine and number of cytosine, so they are same. For example, when you think about human DNA, adenine is you can say 30.4% and corresponding thymine is 30.1%. When you think about cytosine, so 19.9% and then guanine is 19.6%. So from this, what he implicated that DNA captures the molecular diversity among species. Now, this is one of the first experiments which we did to understand this composition of DNA. And then in the composition, when we think about the structure, at that point nobody knew whether that is what is the structure of the DNA. And apart from that, at that time of point, people did not knew that DNA is the genetic material. So there were hypotheses. Does this, the DNA molecule is the genetic material, or is it the same material which is basically passing the information from generation to generation in the organism? So basically, there is can basically at this point some puzzle, when you think about, some puzzle is solved, but then most of the puzzle is still remain to solve. So, people were very interested to know what is basically, what is this DNA molecule. And whatever the experiments are happening, are they, confused or not? So, the key questions over here at that time point was, is DNA indeed the genetic material, and what is the structure of the DNA, and how does the structure account for its genetic material? So these are very key questions, and in order to understand these questions, in order to solve this question, you need to have some kind of experimental data. And the first experimental data for the DNA was of the structural data, you can say, that was that was received applied by Rosalind Franklin, and she got this beautiful X-ray diffraction pattern over here. And this is basically from this pattern what she concluded that basically she could not get into exact model of the DNA, but she could conclude that the DNA has sugar phosphate and basically the sugar phosphate are outside, and the rest of the things she could not figure out how exactly the interaction between nucleotides should be happening, what is the interaction between the nucleotides, to whether how their tracking happens and all those. So basically that kind of information she could not deduce. Only inference she could make that sugar phosphate are outside. And then from there it's a mystery, you can say. The same mystery as Higgs boson. And maybe 19 years back or so, you might have heard about the Higgs boson, and there were a lot of experiments happening in the particle accelerator, which is located at CERN in the Europe. And so basically there were hypotheses about Higgs boson, existence of Higgs boson, whether it's a fundamental particle or elemental particle. And in order to basically you have the hypothesis, and in order to have that hypothesis correct or wrong, you have to basically set up an experiment, and this experiment will verify your hypothesis. In this context, when you think about Higgs boson, we basically actually don't see the Higgs boson, but then with the particle accelerator and all those things, you might be able to see the consequences, how these two particles are interacting, what could be the the end products, and on that basis you will make your observation, and the observation, whatever you do, that will verify your hypothesis. So there is a process to it. So same thing basically over this data diffraction, and from diffraction how do you get to the model of this DNA? And then basically the Watson and Crick, these two physicists came and they tried to understand the structure. They wanted to get the model right. So again they use this Rosalind Franklin's data, but then the interesting thing is that to solve the structure of DNA, Watson Crick got the Nobel Prize, and I think personally I think even Rosalind Franklin should have been awarded Nobel Prize, but then back in days whatever happened basically not. So the important point over here is the Watson Crick. They concluded that, or they came to conclusion that DNA is a helical structure, and width of this helix is 2 nanometer, or you can say 20 nanometer, 20 angstrom. And the spacing between the two bases is about 0.34 nanometer, or you can say 3.4 angstrom. And each helix turn there are 10 bases. And basically one of the most important, I would say, DNA has 2 strands, so it's a double helix, you can say, which goes parallel to each other. So this is how they model it, and this model over here you can see this is James Watson and Francis Crick, and the model built by them over here. So, and this model basically changed the biology, how they understand the biology, how the genetic information, because at that time we knew how this the DNA is the genetic material. But then even though they proposed this model, and this proposed model basically accounts for the Stargardt's observation. So if we go back and you can see the Stargardt model says that amount of adenine and thymine and amount of guanine and cytosine they have kept. So when they Francis and Watson Crick and they got the model, and what they mainly they proposed these three distinct points. So the DNA is double helix, and the width of this is 20 angstrom or 2 nanometer, and there is a base pair interaction between this double helices, and that interaction is basically hydrogen bond, and this hydrogen bond happens between thymine versus adenine, guanine versus cytosine. And this is what exactly the Sargov suggested, that there is a equal content T versus A and G versus C. And on top of that, when what they made is basically DNA is double helix, and the double helix, what happens is the helix has equal direction, 5' A and 3' A. So with this helix, the one helix is running 5' to this 3 point. The complementary, the other helix or the other DNA will have exact opposite direction, which is basically 3' and 5'. So what exactly over here, what we are saying here, let's say you have only a single strand of this DNA, and you bring the complementary strand which is exactly running opposite, so that you can have genetic information on a single strand. But in terms of the stability, the DNA will have a double helix form. But we are not going to discuss the stability of DNA at this moment. So now, when they try to basically come up with this model, what they see is, first thing what they see is this is basically parental molecule, and the parental molecule will have two complementary strands of DNA, and each base is paired by the hydrogen bonding with the specific partner, A with T, G with C. And this is how it would look like. Again, there is a directionality over here. And then the two DNA strands are, if you separate them, then each parental strand over here can now can be served as a template for the new complementary strand. And if you think about the new complementary strand, it would be looking like this. So over here, the new complementary strand. So the dark blue representation is for the parental strand, and the light blue is for the daughter strand. Now, when you think about this, and you have this parental molecule, you separate them, and you bring this complementary. What you see is the complementary is almost same as the parental molecule. So this CG, CA, AT, and GC. Again over here it's AT, CG, CA, AT, GC. The base pairing is same as the parental molecule. So this particular model, what was proposed by the Watson and Crick, how the duplication happens, how the complementary strands, the parental strands can serve as the template for the complementary strands. That particular model is called as semi-conservative replication model. And there are other models at that time. The other model was conservative model and the dispersive model. So now, when we think about these three models, even though the Watson and Crick they proposed this model, semi-conservative model, but still there was no experimental data to it. Even though, so they got the diffraction pattern. From the diffraction pattern, they made some conclusions, but experimentally it is not tested. And then come this another duo, Matt Nelson and Frank Stahl, and they designed a set of experiments. In that, basically, in their experiments, what they did is they wanted to test the DNA replication model, and over there they used bacteria grown in media of heavy isotope of nitrogen. So this is the experimental part. So what they did is, first the bacteria was cultured in N59 heavy isotope of nitrogen. Then the bacteria was transferred to the N14, the lighter isotope of the nitrogen, and then basically they got the DNA sample centrifuged after first replication, and subsequently the second replication. In the first replication, they saw only one band, and in the second replication, they saw two bands. The first band over here, you can say it's composing everything, and in the second band, so basically second replication, what you see over here, you have two bands. One is less dense, other is more dense. Now, when we think about this 7 helix model, the conservative model in the first replication, it should look like this. In the semi-conservative, it should look like this, and in the dispersive model, it should be. So basically, I'll just explain this in a bit. So again, over here, the light blue that shows the isotope from the 14 nitrogen, and dark blue that shows the 15 the isotope of 15 nitrogen. Now, in the conservative model, what it claims that the two parental strands will reassociate after acting as a template for the new strand, and thus restoring the parental double helix. In the semi-conservative model, the claim was two strands two strands of the parental molecules will separate, and each will function as a template for synthesis of new. And that would basically give us, this kind of structure of secondary replication. And a dispersive model simply says that each strand will have a mixture of both, the mixture of N14 and N15. So in the single strand over here, you can see at one patch you can see the light blue, and the other patch you can see dark blue. And then the second replication will have also same combination. So these are the three models. Now, when we look back into this experiment, the first replication you have only single band, and the second replication you have two bands with after synthesization. Now we'll try to get that thing right. So what would happen is, since if at all it would have been conservative model. So again, the conservative model, the first replication should have two separate, and then in the conservative model, the parental will retain and the complementary will have a difference. And in the semi-conservative, there would be pairing, and basically one strand will serve complementary. And in the dispersive model, basically you have the combination of N16 and N14 within same strand. So with this, whatever they observed, and the observation was in the first replication they got only one band. If there is a one band, then the conservative model will have to be negated. So the conservative model does not it does not exist. Then during the second replication, you see there are two bands. So at the first replication, you can consider semi-conservative model and dispersive model. And the result of the second replication, what you see, since there are two bands, and let's say if at all there are two bands, then in the dispersive model that would have been only single band. Since there are two bands, which means the semi-conservative model has to be right, and this will have the two bands will have more dense and less dense objects, or so this less dense is basically from the N14 and more dense is from the N15. So that is what our mixture of the complementary strands with the N14 and N15. So that is how the this semi-conservative model was confirmed by Messerschmitt and Stoll experiment. And with that, I would like to stop here and read it next question. Yes.
Sir, could you explain the basis of conservative model, like again, what the stands for?
Okay. So the assumption of conservative thing. Now we have to basically get back to the conservative model. What does this conservative model say? And in the conservative model, what it says is that you have the parent cell which has now you go back to this experiment. The parent cell over here that is basically isotope, which it has a heavy isotope of nitrogen, that is N15. So which means it should have the two strands with N15. Okay. And what it says that during replication, the parental gene or parental DNA will combine back into its own format. Okay. So the two parental strands will reassociate after acting as a template for new strands. Okay. So that's the basically assumption of it. And if that assumption would have been here, then I should get two bands. The first band should be for the the N14, which is less than, and N15, which is more than. But then during their experiment, for if at all this model is correct, in the first you should get two bands. But then you have got only one band over here after the first application. And that's why the conservative model was not accepted. And then you have two possibilities, semi-conservative and dispersive. And since you have here two bands in the second application, that would basically give us possibility, or that is clear indication that this semi-conservative model is correct.
Sir, I have one small doubt. Like, what do you mean by when you say the DNA in the 14, like the 14N solution? And
this is isotope, right? The nitrogen. See, I mean, you have heard about uranium, right? Uranium, you have the different isotopes.
Yes.
One isotope is basically nuclear. It doesn't mean anything. 235 basically, and.
So I got that. I'm just asking that what do the different color DNA mean?
Different color DNA means, see, okay. For that we have to go back to this, particular structure.
Sir, in not in that slide. I mean the light blue and the dark blue DNA you showed.
Yes, I'm coming to that.
Okay.
See, this when you have the medium and when you provide the nutrients to the bacteria growth. So basically, let's say in that particular growth, you have all isotopes with N15. Then what would happen is all this nitrogen over here, that would be N15. So the parent cell will have all this nitrogen as N15. And when you basically from here, so when you transfer this bacteria, so this all bacteria will have N15, and when you transfer bacteria to the N14 medium, so now in this medium you have normal isotope of nitrogen, which is abundant. N15 is not abundant, but N14 is very abundant, right? So now when you get into this medium, because bacteria needs some food, right? And that food is nothing but a medium. Okay, that is what I want to say. The medium is the bacteria's food. Okay, and in first case, when they start, the medium has only N15. Now you imagine this N15 is being transferred into the medium which has only N14. And then you isolate the DNA. And on that basis, you make the claim. Does that make sense?
Yes, sir. Sir, I have one more doubt related to previous week's lecture.
Which lecture?
Previous week, like we had that in tutorial also we had that Abbe diffraction limit about
Which that. Which one?
ABVE Abbe diffraction limit. In that, the formula was given as lambda by 2 n sin theta, where n is the refractive index and theta is that angle. So, sir, the resolution power, what we were taught was 0.61 times lambda divided by something. So, is that a bit different here, or is that the same thing that we are taught in?
Yeah, okay, I want to understand what resolution you are thinking about.
So,
Because
like,
that resolution is about, the image. And this resolution is different. I mean, I want to understand which resolution you are talking about. In context of the image, in context of the microscope, that resolution is valid, because when you think about the light microscope, there is a diffraction limit, and that within that diffraction limit, two peaks can be distinguished, and I think that is what it says. And I think it basically starts from the Rayleigh's criterion, if I'm correct. But in this, if you're talking about this particular experiment, which experiment? I mean, what is the other experiment you are talking about? That I want to understand.
Sir, I just wanted to ask about in the microscopy and all slides were there. There it was written that whenever we are seeing an cell or something under the microscope, then we can't observe the two objects distinctively if we don't have within that range. Like if they are apart from greater than that particular range, then they are distinctly visible, or else not. That's a limit.
I'm not getting this question. Probably, I have to look into that tutorial slide, because the tutorial slide is made by the CL. So, was it first tutorial? Average diffraction limit B is equal to lambda divided by 2 and sin theta lambda. Okay, that is okay. Yeah, can you tell me again? I mean, I just want to understand this question right. Not able to get this question.
Yes, sir. What we had learned in physics, let me start with that whenever we are observing something under a microscope, we have a particular limit to how much we can resolve between two different.
That is the diffraction limit.
Yeah. So so here the formula was given a bit different. Like there was a missing factor of 0.61. I just wanted to ask, is it different here, or is it?
No, I think they are related. If you are asking me that, I think they are related. I think what you say, that is classically it's called as Rayleigh's criterion, and this is other difference. I think they are related.
Okay, sir. And sir, one more question. Like there was a topic of super resolution microscopy. And it was in addition to this topic only, that how can we resolve two different particles which are in a limit which is smaller than the limit for that particular wavelength. Yeah. So there was a concept of fluorescence objects or something like that. I didn't get that. Can you explain?
So what happens is, over there, because of the the thing is that you have to over there you have to consider many, because too much technical details involved over there. The first thing, the fluorescence, that can be basically when you think about detectors, these detectors, I mean, this is not a thing. This is now you have the pixel. And the pixel basically, you can have, let's say one pixel is there, that can be further divided in terms of the localization where exactly that event is happening. And, the one pixel can be subdivided into 4. That is, and then, let's say each pixel, there is some kind of quantification to that, and on that basis, you can have the subpixel. And with that subpixel, you might be able to localize the signal, and with the localization, you will get little better resolution. So there is it's a little different, I would say, approach over there, and each basically fluorescence molecule has it's a different color, and because of that, the localization is easy, and that is where because you are basically neglecting any other, you can say, light. You are, basically considering, you can say, very narrow wavelength. and that particular color associated with that particular color, and that way it is easy for the localization of the signal, and that's why you can go for the superimposition. I hope that I hope I have answered your question.
Yes, sir. Like, still it's a bit unclear, but I got what you meant by localization and all. So that was clear.
Yeah.
Okay, thank you, sir.
Yeah. Yeah, next one.
Sir, I had doubt in the first application and second application results. So could you explain the part again?
Yeah, sure.
Say in the 14 and 15 MC.
Yeah. See, again, what we say that there are three models. At that time there were three models, and the model proposed by Watson and Crick, that was semi-conservative model. And in the semi-conservative model, the proposition was like this: the parental molecule of DNA that would separate, and after separation, each strand will function as a template of synthesis for new complementary strands. So over here, the light, the light over here, the light strand, that represents the N14 isotope, and that is being complementary. Your first, the parent cell that has N15 level, right? And the parental cell, the parental DNA is getting split into two strands, and each strand is being served as a template for the complementary strand. And the complementary strand, since the growth is happening in the N14 medium, that's why the light blue color is there over here. And this is what they propose, right? And when we think about this particular experiment, which is done by Michelson and Stahl over here, so this first, in the first replication, you got only a single band, and in the second replication, they got two bands, right? So now coming back to this, other model, the conservative model. The conservative model, what that says is basically the two parental strands. So basically these are two parental strands, right? So that will reassociate after acting as a template for new strands. So what happens in between, I don't know, but then what this model suggests that after having this template, the template will have its own double helix, and after that, after serving that as a template, this parental DNA that will again reassociate, and that will form its own, basically own double helix. And if this model is correct, then what would happen is, in the first replication, I should have two bands, and in the second, again, I should have two bands. But in the semi-conservative model, if that is correct, what I'm getting is, here basically I'm getting a mixture of N15 strand and N14 strand. So that would give me only single band. In the dispersive model, the patches are here. Some patches for N14, some patches for N15. Again, in this model, when you think about the replication, you will get only single band. So if I have to consider after first application, I will basically, since this experiment gave me only single band after first application, automatically I will negate this conservative model. I will say that the conservative model does not exist. It's not right. And the right model could be semi-conservative or dispersive model. Now, but again in the second replication, I'm getting two bands. And in the dispersive model, that would have been single band itself, because it is a mixture on the same strand. And if at all I'm getting two bands in the centrifugation, what does that mean? That simply means that one band will be having lighter isotope, other band will be having higher isotope also. So that's where this semi-conservative model was correct. Did you get it? Mohit?
Yeah, sir. I understood it. So I had one more question.
Mm-hmm.
So with the ATGC base test, so
Mm-hmm.
why is the percentage A and percentage T not same? Why is there a bit of
Percentage A and percentage?
so because A and T are connected and C and G are connected, so the percentage is
A and C are connected and C and G connected, yes?
yes, sir. So the percentage of C and G have to be same because
Yes,
they're complementary pairs.
yes.
So but in the data of the percentages, there is some difference.
Yeah,
So could you explain?
obviously, there is a experimental data, the experimental error. These are errors, but then when you think about 1960s, these are astonishing number, I would say. It's very close number, 32.8, 32.1, 29.7, 29.1. They are extremely close. You think about that error, and the observations are astounding. It's just amazing. Roughly what you can say that there is no equal content of adenine and thymine, equal content of guanine and cytosine.
Okay, sir, I got it. Sir, also I wanted to ask about the quiz. So will it be tomorrow or
next tutorial?
Next tutorial.
Yeah.
Sir, also how many questions will be there in the pattern of the exam? Could you just brief on that?
So basically, there will be 6 questions, 6 to 7 questions, and out of that, I will take 5, best question answer.
Sir, objective or subjective paper?
Not subjective. It's objective conducted through Moodle.
Okay, sir. Sir, it will be of how much time? Sir,
10 minutes.
10 minutes only. Okay, sir. Sir, also, will the next lecture will be a part of the quiz?
I will announce all those things.
Okay, sir. Thank you, sir.
Yeah.
Excuse me, sir, am I audible?
Yes.
Sir, I had a bit of a technical doubt. So I registered a day late, and I guess as a result of that, I haven't been added to the TA.
Sorry?
I registered a day late for the semester. Right, and I guess as a result of that, I haven't been added to the TA team from Microsoft. The team for
Ayushman, okay. You are from D3 batch, which, what is your major?
Sir, I'm from engineering physics.
Which year registration?
From a fresher.
Engineering physics.
I'm in the first year.
I will look into it, okay?
Okay, thank you so much.
Yeah. Yeah, so again, what I'm trying to say is, if you are not getting any, so you just have question. Ayushman Sinha, right? Ayushman Sinha. Yeah, there are a couple of, late registration I have to assign them, and as I said, at a times, the data is sometimes is not updated. It gets a little late, and it's very difficult, one student coming and one student, basically, I have to collect data again and again, and that is the difficult part because most of guys have assigned these rooms and all those things, and now I have to make more adjustments. So that is why there is some delay, and mostly the data is not at all updated same time because I have to every time I have to download the data, and let's say I download data today and tomorrow there could be difference. So that is the yeah, I'll try to add here. Next one.
Yes, sir. We have. I will be able to view the test through laptop, or there will be some software installed with mobile and some Moodle only.
That's the software. But if you have only computer, that's fine because what you do is you will check the screen, and there will be you will operating only Moodle, and that screen there will be screen recording that you have to submit in the respective year room. So one single device should be fine.
Okay, so that will not be able to see because the maximum will be slightly up. So.
No. You can allow, when you go for screen recording, you can allow the webcam also. So there are, I will, basically give the guidelines.
Okay, thanks.
Yeah. Next one.
In this slide only, the percentage of adenine is always greater than the percentage of thymine. So, is it possible that while performing the experiment, the adenine molecules from LPP has released into our data, and that's why
HTT is difficult because the extraction process, it happens to the centrifugation. So HTT is a single molecule which is very light. DNA is longer chain, which is longer chain is basically and will have then the molecular weight or weight difference. So
okay, so all the errors are the same.
there could be some errors, and I don't know the particular errors from where it got, and maybe you just think about it. Even today, you might not get that much accuracy.
Yeah.
I doubt it. It's still there could be some other components which I really don't know. But roughly, they account, the 24 and 23, and there are some mutations also, right? I mean, there could be some different pairing, and that different pairing, most of the time, A will be, basically having the base pair with the T. That doesn't mean that other pairing is not going to happen. Other pairing could be happening, but that other pairing will have different layers of structure. So those are, the structural differences.
Yes. And I have another question. So why did we choose our isotope as the N15? I mean, we could also choose as oxygen 18 or carbon 14.
Yeah, that would have been done. Yes. Even that time, that would have been done. Yes.
Because it had to produce the same observation.
Yeah, it would have given same observation. Yeah.
Okay. And
provided over here, I think only problem would have been, you have to get account for this phosphate also. But when you think about nitrogen, then you don't have to account for the phosphate. But this is a little bit challenging in terms of the chemical synthesis because all these oxygen has to be isotopically labeled. And that's where they might have thought, okay, let's go with a simple system, either C13 or nitrogen, and maybe at that time, getting N15 was easy, and that's why they might have thought about getting into N15. Okay, if there are no more questions, then we can stop here, and I think the next lecture is going to be last lecture. Thank you. Okay, yes. Yes.
So, will we be getting a mail regarding the entire details for the course?
I will announce it, right?
Okay.
I said I will, I'll let you know.
Okay. Thank you.
Yeah. Okay.